We're moving on now to talk about proteins, the third of the four types of organic molecules we're going to cover. And first up to talk about proteins is that proteins are a type of molecule that are found all over in your body, and they perform lots and lots of different functions. They do everything from sending messages around the body in the form of hormones. They serve as support in your body in the form of being part of what makes up your bones, your ligaments, your tendons, your muscles. Proteins, a subgroup of proteins called enzymes, are what do things like catalyze reactions that help you digest your food, or convert one type of molecule into another type of molecule. They're used to move you around, such as in muscles. Proteins called antibodies defend your body against infectious agents like bacteria and viruses. And then we have lots of different types of proteins that are found on the surface of cells that move molecules into and out of the cells. Proteins in and of themselves are simply long chains of amino acids. When I string together lots of amino acids, I create a protein. Now, let's talk for a little bit about what an amino acid is. Amino acids are one of the four types of organic molecules, which means by definition they have to have at least one carbon in them. They always have a central carbon. They also always have at least one hydrogen bonded to them. They also always have something called a carboxyl group, C double bonded O, and also a C bonded to an OH. And they have something called an amino group, which gives them their name, which is a nitrogen bonded to two hydrogen. Lastly, they have something called an R group. And that R group can be any number of different molecules, from something as simple as a single atom of hydrogen up to very complex molecules. Depending on what R group you have present, you will have any number of up to 20 different commonly seen amino acids that we use in our body. And depending on that R group, the amino acid can be hydrophobic if it really doesn't have any oxygen bonded to hydrogen present, or it can be very hydrophilic if it does have phosphates in it or lots of oxygens bonded to hydrogen. We join together amino acids to make long chains called polypeptides using that same process we've talked about before called dehydration synthesis. Okay, same process again. Remember, you take the OH group off of one amino acid, the hydrogen group off another, join them together to make a, here we've got two single amino acids. We join them together to make a dipeptide, meaning two amino acids, and a water is given off. The reverse reaction, again, is hydrolysis. We use that water to split apart the two amino acids. A peptide bond is the bond that forms between two amino acids when we perform dehydration synthesis. Now, proteins come in lots of different shapes. Proteins tend to be very long chains of amino acids, and what's a critical thing you absolutely need to know about proteins is their shape has a direct correlation on their function. Okay, their shape and their function are intertwined. So a protein must be folded correctly into its proper final shape before it can function correctly. Now, there are four levels of structure that help to determine the final shape of a protein. The first level of structure, which is called the primary structure, is simply the sequence of amino acids. Okay, we have 20 common amino acids used in our body, things like leucine, isoleucine, glycine, glutamic acid, aspartic acid, tyrosine, phenylalanine. There's all kinds of them, valine, serine, all of them. The order in which we join all those amino acids together determines the primary structure of that amino acid. Okay, the secondary structure of the amino acid is determined by either the fact that the long string of amino acids forms a coiling helix called an alpha helix, or they can fold together in kind of a zigzaggy format like you see up here in this picture, and that's called a beta pleat. Some books call it a beta sheet, but it's the same thing. That's the secondary structure. That secondary structure, the way we get that folding in coils or pleats, is determined by hydrogen bonding between amino acids. The tertiary structure is basically interactions between the R groups. So when the R groups interact with each other, they may either be attracted to one another or they may repel each other. And the end result is we can get, for example, you could have an alpha helix at a right angle to a beta pleat. That might be one tertiary structure. Or you could have an alpha helix parallel to a beta pleat. That would be an example of a different tertiary structure. Again, it's because of the interactions of R groups. Lastly is quaternary structure. Now, quaternary structure is not found in every single protein. Only proteins that have two or more separate polypeptide chains can have quaternary structures. Okay, so for example, hemoglobin, which is the molecule inside of your red blood cells that transports oxygen for you, it consists of what are called 2 alpha chains and 2 beta chains. Those are 4 separate chains that have carefully folded themselves together in such a way to form a functional molecule. Because there are 2 or more separate polypeptide chains that have folded together, we have quaternary structure. Last thing I want to talk about are some of the protein properties of proteins. Proteins can be kind of divided into 2 general categories. There are what are called fibrous proteins, and they're called fibrous proteins because they look like long fibers. And these are also sometimes called structural proteins because their job is to provide structure to a cell or to provide structure to a hair, okay, or whatever it might be. They're there to provide structure and support. Generally speaking, these tend to be hydrophobic. Okay, you have long fibrous proteins inside of your bone called collagen, and that collagen is a fibrous protein that lends structure to your bones. That collagen is hydrophobic, and it's a good thing it is, because if it were hydrophilic, it would dissolve in the presence of all the water in your body, and your bones would literally dissolve away to nothing. By comparison, there's another group of proteins that we call globular proteins, like this guy here. Globular proteins, like their name suggests, usually are very blobby in shape rather than being long and string-like like fibrous proteins. Globular proteins usually are usually enzymes. Okay, and enzymes are a special subset of proteins that catalyze reactions. When I say catalyze simply means that they speed up the rate of a reaction without themselves becoming consumed by the reaction. These guys tend to be very hydrophilic. They dissolve readily in water, so that makes them different from fibrous proteins. Some proteins, some types of globular proteins, are a special group we call chaperone proteins, and these guys are critical in helping other new baby proteins fold into their final shape. And remember, again, we got to get the right final shape in order to perform a proper function. Prions, which if you've ever heard of them, are a type of chaperone protein. Prions are the type of protein that we think causes one version of a prion we believe is responsible for mad cow disease in humans and in cows. In humans, mad cow disease is called Creutzfeldt-Jakob's disease, but it's the same thing. We think it's caused by a rogue type of chaperone protein that's misbehaving. Now, last thing I want to mention about proteins, and this is a really important thing to know about them, is this process called denaturation. Remember, I told you that the shape of a protein is absolutely critical to its proper function. If anything happens that changes the shape of the protein, the function will usually stop. Okay? Now, one thing we know about proteins is anytime I attach something to a protein or I remove something from a protein, it can change shape. Sometimes that can be a good thing. Okay, in fact, that's how we activate and deactivate enzymes, is by attaching molecules or detaching molecules from them. But if we manipulate the environment of a protein such that the change in shape that occurs is a really drastic change in shape rather than a small change in shape, we can get this process called denaturation. And in denaturation, we permanently lose the functionality of the protein such that it's damaged and there's no reversing the damage. Two ways in which we can denature proteins is by altering the pH at which an enzyme functions. Now, every enzyme has an optimal pH at which it functions best. Some enzymes function at very acidic pHs quite well. Other enzymes do not function well at an acidic pH. They actually denature. Another thing we can do is drastically heat up a protein. Typically speaking, higher temperatures cause denaturation of proteins. Every time you fry an egg in a pan and you watch the white of the egg go from being very clear to being white, you are seeing denaturation occurring with that protein. We're going to learn more about proteins later on in terms of some different functions that they perform inside the cells, but this just gives you the basic framework. You should also review about proteins in your textbook to make sure that you've reinforced this information. Our last part of this lecture that I'm going to see you on next time is on nucleic acids.